And now, so I will welcome Hilal Lashuel, professor at the EPFL in Lausanne, that is going to tell us about post-translational modification again, and how this can impact the behavior, the proteostasis of exon 1.
Thank you very much for inviting me again and giving me the opportunity to share with you some of our recent work on trying to understand the grammars of Huntington aggregation and inclusion formation. I will also, for the sake of today's talk, speak a little bit more about our effort to try to develop models and approaches to bridge the gap between the simplistic models that we have from in vitro systems and the complexity of tau aggregation, of Huntington aggregation and pathology formation in neurons. So as we, let me see if I can, this work, okay. This is sort of what we know. We know from what you've heard from the previous talks and pathology, we know quite a lot of the pathology of Huntington. We know that in Huntington brains, we have these inclusions that are formed in the nucleus and the cytoplasm. And we know that these are composed, from a number of Huntington fragments, including full-length Huntington protein. What we don't know actually is which of one of these fragments is the primary initiator and driver of pathology formation. We heard earlier about exon 1. As you will hear from my talk, I believe that other N-terminal fragments of Huntington also play an important role. The missing link, in my opinion, is sort of understanding what is the mechanism of Huntington aggregation and inclusion formation. Realize that I'm using these two words. And how does this process contribute to neuron loss in Huntington? And what is the structural properties of these Huntington aggregates in neurons, and how do they interact and influence the environment inside the cell? Addressing this knowledge gap is not only important for understanding the mechanisms underpinning this disease, but also for developing therapies and for developing HD-relevant disease models. Now, these are the two stories I would like to tell you about today. One addressing this question is which form of Huntington is actually responsible for inclusion formation, and does it matter? And the second one is from a recent studies in our lab using correlative light electron microscopy and mass spectrometry studies to try to understand the mechanism of Huntington inclusion formation in neurons and how they contribute to Huntington toxicity. So we know from studies from many of the people who in many labs that are represented by people here that small changes in the sequence of Huntington within exon 1, either in the polyQ domain or the N17 region in the form of the length of the polyQ repeat or the specific PTM significantly modifies the aggregation and toxicity of Huntington in animal and cellular models of Huntington disease. What we don't know is whether the effect of these sequence changes is actually mediated by their effect on the full-length protein or on a fragment of the Huntington protein. And this is a hypothesis we sort of sought to test. When the cryo-EM structures of Huntington came out, we were all very excited because we thought we'll get a lot of information from the structure. Unfortunately, the regions we're interested in, exon 1 and other flexible N-terminal domain, did not appear in the structure because they were too flexible at that time. Nonetheless, we thought that maybe deciphering the cryo-EM structure of the wild type and the mutant protein could be informative. And you just heard the talk of Stefan, where they showed basically that the structure of Huntington containing different polyQ repeats is actually identical if you overlay here. And therefore, we thought probably one of the reasons is when Huntington is bound to HAP40, the protein is too stable, and that most of the effect of these sequence changes in the protein could be exerted on the flexible, sort of free state of the protein. And therefore, we asked the question, which many people have asked for years, but the experiment was never done, and that is, can we induce the full-length protein to aggregate, or does the full-length protein aggregate? And to address this question with the help of CHDI, we got proteins containing different polyQ repeats. Here is full-length Huntington protein, and you can see it's predominantly, it has these globular structures. And I had challenged one of my research associates in the lab early, Idris, if he can find a way to induce Huntington to form fibrils, and he couldn't. And this work is now led by a talented PhD student, Enzo Moro, who's here in the conference. And in short, we've tried very different conditions, changing pH, temperature, incubating time. We can never induce full-length Huntington to form fibrillar aggregate. It does form high non-fibrillar aggregates that can be sedimentable because of the large size of the protein. And therefore, we hypothesize, it is known that if you take a small, highly aggregation-prone sequence, attach it to a large protein, that you can induce it to aggregate, for example, A beta to JFP. But in the case of Huntington, the size, the exon 1 only constitute about 3% of the protein. So we believe that the large size of the Huntington protein present a very strong steric hindrance for the full-length protein to aggregate on its own and form fibrils. So at the level of exon 1, we know there is a direct correlation between the polyQ repeat length and exon 1 aggregation. We also know that from studies that we have done in our lab and summarize here, that all the post-translational modification in exon 1 either have no effect or inhibit the aggregation of the protein, with exception of two, with exception of ubiquitination, which seems to promote oligomerization. The idea, and we know from in vivo studies that sort of modification that inhibit exon 1 aggregation results in a protective effect in animals. So at the level of exon 1, the effect we see in this sequence seems to be predictive of what we see in the models. And this basically led us to hypothesize the following, that proteolytic processing of full-length Huntington may actually be required for Huntington aggregation inclusion formation and could be a parallel mechanism to the alternative splicing mechanism proposed by Jill Bates and colleagues. And to test this, we needed one, you need to know what the enzymes that are responsible, and two, which polyglutamine-containing fragments are responsible if they're generated by proteolysis. To test this concept, what we did is with Enzo is we basically looked for proteases that can cleave Huntington protein, but do not cleave within the exon 1, and can cleave in neighboring sequences that give N-terminal fragments similar to the ones that have been reported previously, for example, around 128, 126. And we found a couple of these enzymes, and the idea that when we cleave with these enzymes, we can produce these different fragments, 167, 128, and exon 1. And then we asked, what we did is to see whether we can generate these, and which one of these can we do this? Does this lead to the generation of exon 1? And can we get aggregation? And what you see here is the effect of upon treatment of the enzyme. We see quickly the generation of very different N-terminal fragment. These are all stained with exon 1 targeted antibodies, and you see the buildup of the exon 1 protein over time. It starts to build up around 24 hours. And you can see when you use antibodies against exon 1, we cannot detect this, but we still detect these fragments, suggesting that they're probably less than 170, sequence below 171. And when Enzo does this experiment, what you basically see is that at early time points, you see Huntington is mainly a globular protein. At 4 hours, we see the formation of these non-fibrillar aggregates, which we know now are aggregates formed by the longer fragment of Huntington. But at the time that coincides with the formation of exon 1, we see the formation of filamentous aggregates. So we can show that through proteolytic processing, release of N-terminal fragment, we can actually get the release of exon 1 and fragmentation, suggesting that this is a possible pathway that could occur in vivo. What we notice is that the longer fragment form different types of aggregates. So to do this, we try to look at the effect of the N-terminal fragment length and aggregation, and we generated different fragments of Huntington up to 586. And what you can see here, for example, in the case of 586, again, the protein does not aggregate no matter what we do. However, if we induce a specific hyperphosphorylation pattern in 586, we can now get the protein to form fibrils. Some of you may know that this fragment actually does not aggregate in cells, suggesting that in this case we have a post-translational modification as a mechanism of inducing aggregation of Huntington. When we tried to look at the length of the N-terminal repeat, we looked at exon 1, and what you could see when you have exon 1, it forms mainly these single filamentous aggregates. If we start to go to longer fragments, we can see that the kinetics of aggregation change, but mostly once we go above residue 104, the nature of the aggregates change. We no longer form these single fragments, we form these sort of dense structure of fibrils. And what Raja Kolo in the lab, is he investigated the mechanism of aggregation of these long fragments. In this case, I'm showing an example of mutant 171. And what you could see is that the protein begins to form these oligomers. But unlike exon 1, which goes from oligomers to fibrils directly, this goes through the formation of these diffuse type of aggregates that are non-fibrillar, that from which eventually grows fibrils toward the end. So these are non-specific aggregates that serve as nucleation site for the growth of fibrils. And what we were able to show is that unlike exon 1, where the aggregation is driven by the NT17 and the polyQ, in the case of this long fragment, aggregation is actually driven by this globular domain, which is a helical domain. It drives the initiation of aggregation into these amorphous aggregates, and the fibrils grow then at the end of these dense structure. And we can show that this is the mechanism because if we take mutant 171, add excess of only this domain, we can actually block the aggregation of this protein in vitro. So we showed 3 different pathways of initiating Huntington aggregation depending on the fragment length. NT17 polyQ dependent, globular domain dependent, C-terminal globular domain dependent, and hyperphosphorylation dependent mechanisms. And what this shows is basically that sequences outside the polyQ domain not only modify the aggregation of the protein, but can also play a key role in the initiation of the aggregation process, and that different N-terminal fragments could have distinct aggregation mechanisms, and therefore a single polyQ anti-aggregation strategy may not be effective to inhibit the aggregation of all N-terminal fragments, and future screening approaches targeting this Huntington aggregation may require the use of different substrates. Finally, I'd like to speak a little bit about our effort to understand the mechanism of aggregation in cells. And this is work that was led by a former talented PhD student, Nathan Rigui, who just graduated recently. And what we were interested is in deciphering the mechanism of cytoplasmic and nuclear Huntington aggregation. And for this, we used 2 different models. We used the HEC293 cells because they form predominantly cytoplasmic inclusions. And then we use primary cortical neuron because through the overexpression of exon 1, we can induce predominantly nuclear inclusions. And we followed, to characterize this, we use a new technique called correlative light electron microscopy. I will explain it. Initially, when we looked at Huntington inclusions in HEC, we see what most of you have always seen, this ring-like sort of organization of Huntington. And it doesn't matter which antibody we use, we saw the same. And to us, this suggested 2 things. One is that either the aggregates are very condensed and the antibodies cannot penetrate the inclusions, or there is something different in the core that is different and Huntington is only in the periphery. To decipher this, what we did is we used correlative electron microscopy. Those who are not familiar with this technique, what we basically do is we label the cells, and then we take the cell that has the aggregate and we section it with electron microscopy, and therefore we can study the structure of the aggregates. And what you can see here is, unlike what people have always proposed, if Huntington aggregate are just inclusions, they're not made. You can see, first of all, that they're organized in a very specific architecture. They have this dense core and a shell around it, a ring-like structure. In high-resolution images, we can see radiating filaments here. But most importantly, we know now from proteomics studies and advanced is that these are not made only of Huntington. There is Huntington aggregates. There is lipids. We can label different types of lipids. There is accumulation of disrupted organelles, including the ER, and accumulation of mitochondria and other membrane structures in the periphery. We find hundreds of proteins, including proteins that are cytoskeletal proteins and proteins that sort of involved in mRNA processing as well as nucleoplasm-related proteins. So it's a much more complex structure, and there is more to Huntington inclusions than just the presence of the fibrils. If we looked at the effect of the polyQ length on the ultrastructural properties of these inclusions, you can find big difference between the 39Q and the 72Q. This ring-like architecture is not present in the 39Q. It could be possible that this is a matter of inclusion maturation, but what is interesting is that the NT17, which is thought to regulate many of the processes of inclusion formation, does not influence this architecture. So if we remove NT17, we still get inclusion that are as complex as the ones formed by the full-length protein. So then we tried to look at what about nuclear inclusions that form in these cells. And we did the same experiments, and what you could see is that the nuclear inclusions are also different. They are composed primarily of these dense fibrillar structures that are compact together. They don't have any lipids or organelle components with them, and they're basically distinct, suggesting that Huntington inclusions in the cytosol are completely different than the nucleus, and that the cellular environment is an important determinant of the complexity, biochemical and ultrastructural complexity of the inclusions. What is interesting is this composition is very similar to what we see now for composition of inclusions isolated from human brain in Parkinson's disease. These are actually same Lewy bodies analyzed by correlative light electron microscopy, and we can show it is now known that these are composed of these complex structures of aggregates, lipids, and organelles. And actually recently, a colleague showed me images of an immature Lewy body that looks very much like this. Okay. So the implications of this is that there are distinct compositions, and which is just distinct mechanism of aggregation, and that the composition and structure of HTT inclusions could be different in different cell types as well. Next, we wanted to see, this was in HEX cells, we wanted to study this in neurons, so we wanted to look at the structure of Huntington aggregates in neurons, and Nathan did a similar study. These are the images of Huntington nuclear aggregation from the brain that have been published. And you can see they do suggest some sort of a fibrillar structure, but it's very granular and difficult to elucidate. In neurons, what we know is that if you overexpress exon 1, we can induce the formation of these nuclear aggregates. They mature in size, they increase in size over time, and we do see an increase in toxicity over time in neurons due to the overexpression and inclusion formation of exon 1. So we did similar correlative light electron microscopy studies, and what you could see is we see very similar granular structures to what's seen in the brain. It was very difficult to decipher by using simple electron microscopy, so we switched to electron tomography, and nothing better than a movie to show this. This is what these Huntington fibrils looked like in the nucleus of neurons. You can see that they're composed primarily of fibrils, similar to what we see in HEX cells, and they don't have that complexity of the cytoplasmic inclusion structures. So in the case of the cytoplasmic inclusion, our study suggests that the process occurs in two phases. The first one is a very rapid phase that we believe is driven probably by phase separation driven by the polyQ domain that forms this dense core. And the second process is mediated by the growth of the fibrils on the surface through monomer addition, and that this dynamic process involves interaction with other proteins and organelles, giving rise to this sort of core and shell structure. We know this because if you were, I can't click this movie, but when we monitor the kinetics of Huntington aggregation in these cells, you can see that you go from no inclusions to inclusion formation and complete depletion of Huntington just within about an hour or so in these cells. Okay, I'm almost done, so it's okay. So in conclusion, what I hope, this is what we've learned from these studies, is that cytoplasmic Huntington inclusions are composed of a complex mixtures of aggregate mutant Huntington, different cellular proteins, and membranous organelles, including endosomal system. Nuclear and cytoplasmic inclusions exhibit distinct biochemical composition and ultrastructure properties, suggesting different mechanism of aggregation and inclusion formation and toxicity, and that differences in the cellular environment and interactome influence the final composition and ultrastructure, which raises the point of the possibility that Huntington inclusion in different cell types could be different. We need more studies to investigate the role and interplay between lipids and organelles and Huntington aggregates, and hopefully in our lab, we're trying to extend these models using longer N-terminal fragments into iPSC from human patients. And similar study, I wanted to point out to this paper from Helen Sable and Jill Bates, in which they've done correlative light electron microscopy and mutant Huntington aggregates in the brain, and their findings illustrate this complexity of these aggregates and their interaction with membranous organelles. With this, I'd like to end by thanking the people who were involved in this work. Nathan Rigua was involved in all the cellular work that I showed in the correlative light electron microscopy. Raja Kola is a postdoc in the lab who did all the work on the N-terminal fragment in collaboration with Idris and Iman. Sergei Nazarov did all the electron tomography studies and cryo-EM of Huntington fibrils that I didn't get to show. And all of the imaging work was done in collaboration with our colleagues at the bioimaging facility, and mainly CHDI for their support. All of this work was supported by them. And with that, I would like to thank you, and I'm happy to take questions.
Thank you, Hilal. We have time for one or two questions. Are there some questions? So maybe I have some, actually. So you mentioned that there is a distinct biochemical composition between the cytoplasmic and so this ring-like structure, actually, I was quite amazed, because this reminds me some experiment I did a long time ago about because when you express short fragment into the neurons, primary neurons, you mostly have nuclear inclusions. Yes. But what we found is that when we were modifying the ubiquitination pathway, we were able to see that type of string structures in the cytoplasm. So that fits with the ideas that you have like several pathways that are probably different between the ones that are in the nuclei and in the cytoplasm. So do you have any idea about given the different composition you have? I mean, did you find again ubiquitination or that type of things?
I think there are 2 points I'd like to make, and just a reminder to everybody. When we think about aggregation, aggregation is not a single product, this process is not a single, we usually think about Huntington aggregates. From all the work we know now, from Parkinson's disease, from Alzheimer's, there is aggregation, and then there is inclusion formation. Aggregation usually involves one protein. Inclusion formation is a much more complex process that involves interaction between the aggregation process and the environment. And we believe that because of the proteins and the organelle, those close interaction, that's what drives the differences in the composition. So wherever these aggregates take place, their transition into inclusion or their maturation will be dependent on the local environment, whether it's the presence of specific kinases or proteases or different interactome.
Okay.
Thank you.
May I have a question?
So we just have one brief question from Eric Van Kerk.
May I have a question? Back here.
No.
There is one here
in the back.
Terrific, thank you.
Yeah.
Thank you very much for that. That was terrific. Paul D'Souza. So I'd like to bring nuclear inclusions back to DNA repair mismatching. Do you have you or anyone in the field, any reason to believe that these inclusions could interfere with or be a driver of DNA mismatching and repair difficulties?
Yeah, I think it's a possibility. I mean, we have not looked at it, but I think it would be something very interesting to look at. One thing that I haven't mentioned is that in all of these inclusions, we always see recruitment of normal Huntington as well. So, anything, this could lead to the sequestration of Huntington and loss of normal Huntington function plus other proteins. But to go back to your points, we have not looked at that. We'll be happy to collaborate with anyone who's interested.
Terrific. Thanks so much.
Okay, so I think we have to stop the session. So, Erich, I suggest that you ask directly to Hilal, okay? Sorry.
Yes. Thanks to the speaker for the beautiful talks and the very interesting discussion. I think it's time to go to the poster session.
Exactly.
Yeah.
So,
Thank
enjoy
you.
the poster session and the break.
Thank you.
Thank
very
you.
much.